Sustainable Development Goals are comprised of 17 goals formed by all United Nations members. They focus on addressing and fixing multiple global issues like poverty and climate change. Goal 14 is to conserve and sustainably use the oceans, seas, and marine resources for sustainable development. The ocean is vital for regulating the climate and supporting biodiversity. However, it faces significant threats from pollution, overfishing, and climate change. Goal 14's second target primarily focuses on the protection and restoration of ecosystems. We will be addressing how molecular biology and aquatic ecosystems is a key factor in maintaining the biodiversity and health of our oceans.
Molecular biology is concerned with how structure and function work at the scale of molecules. Understanding this process can assist us in conserving marine biology by helping us both to protect and restore aquatic systems. Molecular biology methods are useful for understanding the fundamentals of biogeochemical processes and for detecting alteration in the fish supply chain. The management tools of genetic diversity monitoring and habitat protection play a central role in global marine conservation strategies. Due to genetic and molecular biology research advances such as DNA-based barcoding and genomics, it is now possible for scientists to track the genetic diversity of marine species. Marine conservation managers can now use this information to monitor the genetic constitution of populations and keep track of their diversity, which can then be used to identify those at risk. Moreover, for species hard to spot, such as many shark species, genetic monitoring is complemented by the environmental DNA technology, which monitors whether a species is present and in which quantities in areas that are protected, such as marine reserves. The knowledge helps us monitor whether conservation works and the important habitats are truly protected.
There are several marine protection strategies having already been researched and implemented around the world by molecular biologists, one being DNA barcoding. This technique involves extractions of DNA segments containing different genes of organisms in order to identify the species diversity in a particular area. By taking samples back to the lab and recording information in a database, researchers can retain data over long periods to monitor the abundance of populations. The early detection of population decline allows for immediate action and implementation of conservation strategies. The increasing threats faced by sea turtles, marine biologists in Indonesia have implemented new and innovative strategies to increase conservation efforts. Along the coast, 4 different sea turtle species have been investigated, analysing the DNA of tissue samples. The short segments of DNA can be used to identify the different species, collating information in a database. By monitoring each species, turtle population patterns have been observed, and in response, a primary focus on preservation strategies ensures the longevity of these critical organisms. Marine protection strategy is genetic tagging. This procedure gives us insight into the specific genes within a cell, enabling conservationists to observe and contrast between species. In South Australia, after seeing a decline in the abundance of the southern bluefin tuna species due to genetic tagging, conservationists have focused strategies towards rebuilding these populations. Using the DNA from the juvenile fish, the tags track vital details in order to identify fluctuations.
Biology techniques have advanced drastically over the last century, with a majority of advancements being made in the last few decades. As technology continues to improve, all aspects of molecular biology techniques will be able to be further developed. This will allow data collected to be more accurate and make processes more efficient, so more progress can be made into protecting and restoring marine ecosystems. As discussed previously, DNA barcoding and genetic tagging are 2 examples of current molecular biology techniques. Both of these techniques have developed quickly and will continue to develop and change with time. Future development for DNA barcoding is the improvement of curated reference libraries. These libraries can give scientists and researchers access to accurate and reliable DNA barcoding data that they can use for their own investigations. Having access to data like this will save time when carrying out research to understand the way species in ecosystems interact, which means solutions to protecting and restoring marine ecosystems can be made quicker. In terms of gene tagging, although the technology is developing quickly, there is still the potential for false positive and negative matches, which means that very particular methods must be undertaken, which can be quite tedious and time-consuming. Future improvement and development for genetic tagging is the development of higher quality technology that is accessible and can assist scientists in getting better samples and reducing human error. Ultimately, all future developments for molecular biology need to be accessible and affordable and make the process of collecting and analysing data more efficient whilst also achieving higher quality results. These future developments will allow our oceans to be conserved and used sustainably and allow for the protection and restoration of marine ecosystems.
Techniques like DNA barcoding, genetic tagging, and environmental DNA monitoring allow scientists to track the genetic diversity of marine species, detect population declines early, and implement targeted conservation strategies. These methods have already shown success in various global efforts. As technology continues to evolve, advancements in molecular biology will further enhance the accuracy, efficiency, and accessibility of these techniques.